Thank you. Thank you for this opportunity. Thank you, Reinhard, particularly. I'm a clinician, and this talk is going to be really quite simple. I hope intuitive, even for those not familiar with the phenomenology of epilepsy or EEG. We just had a fantastic introduction from Danny Bassett. And the methods are going to be very simple in comparison to the sophistication of what we've heard all week long. It's not common for a clinician like me to really be in the company, in such exalted company. But I'm very grateful, and hopefully this is a metaphor. The metaphor modularity will appeal to some of you in the field of epilepsy, and certainly I think this is a useful one for us in our field. So with that, I didn't know what Danny was going to say, so some of it will be like a dumbed down version of what maybe she has said already. But just to introduce the idea of modularity in a very simple, very intuitive way, at least when you come to it fairly naively. We're all aware, of course, of gross structural modularity in biology, in our beings, in the nervous system. We have two hemispheres, and we have a central nervous system, and then it is sort of it has another structure caudal to it called spinal cord, and so on. So we are familiar with that. There are asymmetries in these. One thinks of modulars, say something that is being patterned in space as maybe perhaps being the same left to right, but there's some asymmetries that are built into it that are really quite profound, which I won't go into, like handedness. So if we had two modules, right and left, or you had a structure, the hemisphere that was broken into two by some process that made one into two, why should there be a left and why should there be a right to this, right? So it's really kind of a profound question. We won't talk about it, but this is just something we observe. I'm just saying, coming to it, these are things you just see. This is the landscape in which you're now going to operate. We're familiar with brain lobes in areas, and there are various ways of defining them. And these are all gross and crude, and these are structural. There's no processing here except for observation. And then, through the work, Brodmann is mostly quoted, but there's several others, where you have gradients in the structure of the brain, right? So cytoarchitectural gradients when you look at it through a microscope. But you can do other things. You can measure things. You can measure the amount of neurons in a cubic centimeter of brain. And in fact, there's a gradient. Most of them are at the back, or there's a big concentration at the back. And for some reason, the more forwards you go, there are less neurons in your brain. You can do the same with things like myelin and so on. In addition to these, we have functional modules, and we got a whole talk on the, how functional modules are defined, their role to structure, and so on. But in a very simple way, structure-associated modules are really quite easy in the first instance to even imagine. So if an organism has to be given the task of moving forward, certainly you need a motor system to be able to do that, and therefore an information processing module in your brain to be able to do that. It's not really difficult to understand of why a motor system module exists. So that's not, I don't believe, is a big problem. It's a problem maybe in some other sense as to how all of that develops and how it all connects and so on. But the very existence of it is not really an issue. Similarly, for instance, if you told an organism was given the instruction, listen, go out and sense electromagnetic waves and make sense of the physical world around you, through that, you would develop a visual system, right? Now, beyond those, there are more subtle modules, and we've heard several already this morning. Task-related modules that come into being when someone is asked to do something, right? And so these may be along the lines of predefined structure, but they may be somewhat outside it as well. We've heard of other modules that are even more subtle, that were only discovered 20 years ago, okay? Modules that were defined through brain imaging, such as the DMN and other kinds of canonical networks, right? Which for some time it wasn't realized what they do, and in fact whether they even exist and why they should exist in the way they do. With all of that broad introduction, we will move on to the world of epilepsy and electrophysiology and EEG, which is also modular. And we've done a little bit of experimentation in our group, try to illustrate some of that, to bring out some of those, ideas with real data, with varying success, I have to say. But we had only so much time, and So those are electrophysiological modules. These are the way EEG behaves over space and time. And finally, of course, and why do you bother? Why should a clinician bother? And why is this relevant? Or why is it possibly relevant to disease? Is that epilepsy is thought to be increasingly a disorder of networks. And that is a word that is sometimes used loosely, sometimes a little more precisely. But I think it's a very useful type of concept. So we can hang all of what we conceptualize about epilepsy in that particular framework, or at least some of it, and see what comes off it. Just another little intro slide. Structure, you see this and you see the stripes on a zebra or the pattern on a zebra, you wonder where it comes from. And long ago, Turing, 70 years ago, proposed this very famous model, reaction diffusion system, which will give you stationary spatial patterns, right? Now, this is still a subject of active investigation as we speak. It's a very simple system. It's got, then the whole counterintuitive thing about it was that you have something that is stable, purely in time, and the reaction. So if the diffusion isn't there, the system will go to equilibrium stably. And if you have just a diffusion term, of course, it tends to iron out all spatial inhomogeneities. But if you put them two together and D1 is sufficiently different from D2, you get spatial pattern. This was Turing's great insight, right? And this is still an active subject. And in fact, it sort of is an allegory. It's metaphor, right? For a lot of things. Now, you can put the same thing in here. And you study anatomy, no one tells you about Turing patterns. But here it is, and this is a structural module, if you like. It's really a very literal interpretation of Simons' near decomposability. You can think of your spinal column as being a nearly decomposable structural system, right? So it gives you the advantage of being both flexible as well as giving you shape, right? We've heard a little bit about all this already. Okay, so that's sort of a general sort of pattern formation and the appearance of gross structure. But coming to the brain itself, you have structural modules, or you have structural, if you like, segregation or spatial patterning that appears, whether you look at it through a microscope with Brodmann, or you look at it in other ways, and you say assay myelin, or you look at connectivity, and we've seen some of that already. Or you have someone doing a task relation, some sort of task, and you have areas of the brain that become active or light up. Or you can do something like this, and I'll talk about this in the next slide or two, I think. Okay, where you have resting state EEG, not a resting state fMRI, and you compute some metric of it. Okay, you compute something that you derive from that, and you realize that in here, which is called temporal hierarchy, the intrinsic time constants of EEG change. So some portions of EEG decay very quickly, so information is lost very quickly. There are other portions of EEG where it decays very slowly. So you might think that the way it decays very slowly are areas where there's lots of processing going on. And where, in fact, it decays very quickly are areas that are purely effector or purely sensory. And all they do is gather information and transmit it elsewhere. And that turns out actually to be true. In the world of clinical medicine, we don't use these words, topology, but we do use the word hodology. And this is our network, by the way. So for those of you who don't know about a clinician neurologist network, this is it. And by the way, this is over 70 years old. So the point is, we might, think of modularity as something that maybe now today, here we are in an interdisciplinary conference. But of course, ideas of this type go back and in various shapes or forms ever since people have begun to think about these things in a systematic way. Norman Geschwind was a famous figure in the world of behavioral neurology. And this is, in fact, the scale at which we operate. This is what, by the way, is relevant. You might think of this as crude. This type of parcellation of the brain into these blobs might be sort of, not sophisticated enough or very simplistic. But this makes sense because this is useful. And at the end of the day, I'm going to mention this in my last slide, so I won't give the punchline away. You have to operate at a scale that is meaningful to you. So this is Geschwind's work, where he says that all these cortical areas. And it's not just the fact that you can have a problem in one area that gives you a symptom. It's also the fact that you have connections between areas. So Geschwind really pioneered this whole area of what are called disconnection syndromes. So in our language, or language of this particular workshop, these might be nodes, those might be edges. And you get disease from problems in the nodes and problems in the edges. Just one slide to say that none of this is been fully, it's not without debate. Debate continues about the centrality of brain areas, what brain areas really mean, and whether there's some dynamism in it, which along the lines of what I think Danny said earlier, but a lot of other speakers have said too, about the fact that modularity isn't something that is fixed. It depends on context. And we heard that I think in Nitin Baliga's talk too, that you have a system here in the middle, and the modular output that you get on the right side depends on what perturbation comes in on the left side. Into my world now. This is what we do for a living. We look after patients who have this disease called epilepsy. We gather data that comes from brain surface. We gather data sometimes that comes from under the skull, over directly on top of the brain. These are safe procedures, by the way. Okay, the benefit is much more than the harm that is done. We hope to do no harm, of course. And these are patients in whom the data are collected from within the brain substance itself. Okay, so this is these are opportunities to collect and study waveforms. And in the humdrum of our clinical lives, we are interested in diagnostic waveforms. We are interested in localizations that are really fairly elementary, okay, to identify. And therefore, we design treatments that are based on our interpretation of how this network behaves. Okay, I hope you can all see this. These are just two penetrating electrodes inside the brain of a real person. This person is sitting having breakfast talking to you, by the way. So they're not sick, even though they've had something of this type done. And there are many of these, and some of this data we saw earlier, we've seen in our workshop as well. And our job is to find out amongst all the various contacts that there are, all the various nodes in the network you like, where the problem is, and the problem is identified by its time signature. This is how, and I'm going to give you a few snapshots of this, and I won't go through the details. It's not really important. I just wanted to let you know that this is what an EEG looks like, and it looks different from channel to channel. That's all I want to say. Okay, so if, for instance, the numbers on the left side are in fact the locations, if you like, okay, of where you are on the head on this particular person, for instance. And the time series that are from that particular region. All I want to highlight is one thing here, that the channels look different from each other. Now, if they didn't, there would be no reason to collect it all over the head. You could just have it from one place in the head. The fact is it's spatially heterogeneous, and you get certain types of waveforms that you don't get in others.
Just question. What is the time scale there? Because there is a characteristic frequency, right?
So correct, indeed it is. Yeah, that's right. So those are the details. I'm glad you asked. You're obviously interested. Thank you. So the big division, the green lines are 1 second. Okay, so it's about 15 seconds long. Okay, so each of these channels is in fact the potential difference between any of these two contacts that you see in that cartoon over there, graphed against time, right? And so this thing goes up and down, up and down, up and down, and that's called the alpha, by the way, alpha being the first letter of the alphabet, of the Greek alphabet. So that was the first historically identified waveform, and that hangs around the back of the head. The fact is, not to go too much into all of that, but the fact is things are different, whether you're in the front of the brain or the back of the brain. And so the content of this particular signal is different. So this just reflects this heterogeneity that I was talking about. You can make things precise, and this is some work we published now, a year or more than a year ago, whatever, where, how do you abstract this? How do you make a sort of an intuitive picture of how brain waves look like from place to place? And so this is EEG that looks a little bit different. This was intracerebral EEG, but, you can parameterize it, and we did this with a mixture of Gaussians and so on of the power spectrum, and then you can parameterize it, create these, turn the parameters into colors, and you get maps like this that look very nice to look at, I hope. And this is in fact the person going to sleep, okay, from the waking state. So it has been done in a way that parameters turn into colors, and this just graphically tells you which area sleeps more. And by the way, we don't all sleep homogeneously, we sleep heterogeneously too. Okay, some parts of the brain. What is sleep, of course, is another question altogether. But if you took a certain electrophysiological metric of how sleepy you are, that metric is different from brain region to brain region. So there are parts of the brain that don't do very much or don't become that deeply asleep by that metric. Here it is. And so you can see you're awake, and we all know that REM sleep, rapid eye movement sleep, you're a little bit like as though you're awake. This is someone in REM in the fourth panel there. Okay, it looks a lot like wakefulness. This is just again to emphasize this, the spatial parcellation of cerebral rhythms, and those are normal rhythms. Now you might think of this also as a normal rhythm because this looks a lot like the alpha, right? That sinusoidal oscillation that someone was interested in, and here it is. And our job is to really identify patterns of this type, and we do this purely through a process of visual recognition. That's what clinical training is all about, okay? And this, of course, evolves, and this becomes what we call a seizure, right? And so I'll come to your question. I do need to say this before I answer your question, which is this is a module, right? So this is something that shouldn't be there. It exists in the milieu. It exists within what we consider normality, okay? And in a patient in whom one of these brain areas is somewhat diseased, you get the development of a spatiotemporal motif, or an organization in time and in space over here, of course, that evolves, and that creates a disturbance sufficient to produce symptoms and obviously cause illness, and we call that a seizure. Right. Go ahead, sir.
Yes, you said that's a module, but so when you showed so you've given us a lot of time tracks, right? And showed us that they're different, but should I be thinking that all of these are tightly coupled, or each track itself is an individual, operating unit? So when you say module, at what level of interconnectivity of all this data that you're giving us should we be thinking about?
I wish you'd come to our working group. No, very great question. Thank you. Of course, they are connected to each other. That is absolutely right. If I go back, not look at the time series, but if I just look at this, for instance, okay, and this is maybe intuitive, very intuitive, right? So you have the surface of the brain, and you have all of these tightly packed contacts, and you're collecting information. Of course, the information from number 36 looks very similar to the information in number 35. How similar depends on what the patient is doing. Okay, there's a little nuance there, whether they're awake or asleep and blah, blah, all of this business. But in almost all situations, 35 is going to look very different from 1. Okay, so there's going to be some decay of correlations, spatially there is. So nearby objects are similar in one state, and they're dissimilar as you move away in general. In general, not always. Depends. What if there's a big white matter pathway that connects those two distant regions? Then you will, in fact, get a correlation that is quite high, even though spatially they appear to be quite distant. But in general, the further away you are, and you can just take even this just simple version of distance, you can't just do it as the crow flies, but you will go up and down, the brain surface like this, and it generally decays with distance, and that decay, the pattern of decay itself depends on what state you're in.
But even what you say that they decorrelate, right? I mean, I could think of coupling a dynamical system, right? So that the behavior over here appears to be very different from the behavior over here, but yet there's a lot of control going back and forth, right? It depends on how you're measuring the correlation.
I agree. Right? It depends on how you assess that.
Yes,
I don't disagree.
how you assess
absolutely.
it. Yes.
Yeah. That's like a field all in itself, actually. But absolutely, yeah. So here, okay, is a bad pattern. We don't, we want to eliminate these and get patients better and so on. Okay, and here are some other examples. Okay, now I just want to come to something completely different, and this is a seizure, by the way, but before I come to that, you're going to see a slide that appears kind of dirty, and I'm sorry, I just wanted to give you the sense of how it actually looks. I should have made a cartoon of it. So here, I'm illustrating, I hope you can see this. I want to illustrate this pattern, okay, here, which I can just tell you is abnormal. So please take my word for it, okay? So here you have in all of these, okay, which is just, don't worry about what those numbers and letters are. This, we just take all these particular channels. There's a particular set of channels that looks different, and that's developing a different kind of dynamic, and this is going to grow. okay, as you watch it in time and becomes seizure, and this is already a seizure at this point. This is all of it. So you have this particular portion of this spatial, of this physical space that is displaying a different dynamic, okay? Sometimes the change in dynamic affects the whole brain. And now this looks bad, but maybe I should have given you a cartoon of this after this. But this is really how the data look, okay? So this is the real world, okay? So this is an actual EEG. This is taken from a different patient, and don't worry, again, these numbers and all of that are a little bit different, okay? What I want to show is this, and this is highly compressed, that's why this looks so busy. This is about 40 seconds or maybe a minute or something like that. Each one before, remember, was just 10 or 15 seconds. Here you have, I hope you can appreciate that there's some bunching. There's the end, as though all of the brain, or all, I should not say the brain, I should say all the electrode activity, okay, is unitarily doing something for a little bit of time, okay, and these are about 5 or 6 seconds maybe, or maybe more, maybe 10 or something, okay. So the entire system goes into one regime. The entire system goes into another different regime. The entire system comes back to the same regime. Flip-flop, flip-flop, flip-flop. This can go on for hours. This is just one other type of pattern. Now, our job, much of training and much of what we do is to recognize patterns like this. We become used to, in the course of our training, dozens, scores, maybe hundreds of things like this. Objects like a zoologist. You actually go there and you identify each kind of beast there is, and you create a diagnosis, right? Or you at least give it a name. And that name, that particular entity then gets correlated with a certain state in the patient, right? Now, by the way, these kind of patterns occur in rather sick patients. And this is called cyclical alternating pattern. And we saw an example of that in our working group too. The question is, there are questions here, right? The questions is, first of all, why does this occur? No one really knows. There are two regimes here, okay? They're visually obvious. Those two regimes are associated with slightly different correlative spatial structures. Okay, so if you took a map of how the brain self-interacts through whatever metric you want, okay, cross-correlation or phase synchrony or anything like that, and you got yourself a mosaic of how the brain self-interacted in this regime, it will naturally be different from how it self-interacts in this. But somehow it seems to go between one and the other spontaneously. It's not clear why it happens. We do know it happens in sick people. We don't know why these are the length they are. We don't know why there are only 2 states. Why not 5 or 6?
Also quick, what's happening to the patient in this moment?
The patient isn't jumping up and down. No, the patient is, there's no change. The patient is quite ill. They're in an intensive care unit. Some of my colleagues have a great interest in this pattern. I'm actually just representing their interest, but in a slightly different way. They're interested in knowing when this occurs and, on the patient side of things. I'm interested, or I'm presenting to you the dynamics, okay, and the more fundamental question behind this as to why these patterns arise at all. And the further questions of why they are as long as they are and why do they alternate and all of this other business. And finally, when the patient gets better, they just go away. This is just one, yeah.
So again, you said there's 2 patterns, right? I mean, sitting from the way back here, it's clearly, something happening and very little happening. But if I look closely at any individual pulse, there's differences, right?
Yeah, there is. Yeah. It's not exact. Yeah, of course. I mean, a state is not one entity. A state is, it depends on what you mean by a state, right? So, you have to in some way, group configurations as belonging to one state, similar enough as belonging in one state, and then they're different enough to belong in another state. Now, for practical purposes, of course, there's heterogeneity even between maybe this and that way, there is. That's probably not as relevant as the fact that as there is a much bigger change happening that is of this type. Okay, so yeah.
I have the same question. I do not know whether this is 5 states and 2 states. I mean, on some level, it looks like 2.
Yeah.
But the other one is that, it is not clear to me immediately why the quiet state versus the active state are more synchronized than the other? Because I don't know. I mean, you said that there may be a different interaction pattern between the parts of the brain, but it's very hard to discern here from this.
Yeah, I don't have an answer for you. Okay, so I can't tell you what exactly is different. I'm trying to highlight. I'm just trying to show you a phenomenon. And the kind of corollary to that, or whatever the addendum to that, is there are a lot of phenomena of this type. And the physician, I think we have one physician in the audience, would know that often, our job is to really take end products of really very complicated processes. You get a clinical signal at one end that you recognize as belonging in some sort of class. And then you kind of see it often enough for you to recognize the next time you see it. And then you realize that there's a kind of certain patient type that tends to produce this. And then you understand the whole trajectory, and you believe you understood the causation, or not. You can never say you understand causation. You understand the correlation between a certain sort of macroscopic phenotypic clinical presentation and its final expression of this type. And I have to say this in the defense of conventional medicine, that sometimes that is enough. If this person gets better because whatever you did, something, get an antibiotic or something, and this goes away, or is it really important to know this? Probably not. Okay. But I'm claiming that there are certain phenomena that we see where it is not incompletely understood, and the answers are consequential. Okay. Just out of interest, I mean, I don't know, maybe most of you know this piece of folklore. It's not actually that dolphins sleep with one hemisphere at a time. People didn't know that. They sleep with one half of the brain. Okay, there you go. I'm glad I've given you some entertaining information, but this is the original Russian paper that demonstrated this. Okay. So the switching behavior, this is just because of switching behavior. So here there's a switch of the whole brain, that there are two kinds of state, and everything changes. And here sometimes when you have a switch between two of the structural modules, right? So isn't these interesting dynamics? I mean, I find this very interesting. When you have, it's not clear what is causing the switch, why there are only two. Okay, in the case of the hemisphere, there can only be two because there are only two hemispheres. But what is it that is driving them? What is it, what dynamic phenomenon underlies them? And how can we trace a path, and what is, can we understand this enough so that when we don't have effective treatments for certain conditions, we can perturb this, and do something about it? Okay, so I think I've already said this. This has been implicit all the way, that when Reinhard asked me to talk about modularity, this is the only thing I could think about that is relevant to me, is that a module in the world of human clinical neurophysiology, which is EEG, is really a spatiotemporal motif. And a seizure is only one example of a very wide variety of motifs that we see. So further addendum to this is this, and this is, I think, worth knowing, especially in the instance of seizure, for instance, or even in the patient that was showing this alternating pattern. They may have nothing wrong with their brains, by the way. They may have liver failure or something, and they're in a coma, and then the liver failure gets better, they get better, they walk out of hospital, their brain waves come back to normal. When they were very ill, their brains were behaving that way. In this case of a seizure, for example, a seizure starts in one part of the brain and spreads to the whole brain. Okay, we all know of people who have seizures, they fall down, they convulse, and so on. But most seizures, in fact, many seizure types actually stay restricted in only one portion of the brain. Sometimes they will migrate, okay, and they will go to other parts of the brain, which were normal. So what does that tell us? That tells us that these motifs that we think of as abnormal are implicit, are kinds of behavior that a normal brain can itself produce, right? So when a seizure goes from one portion to another, and the kind of behavior, the motif that starts in one place is recapitulated, or in fact is transmitted, or expresses itself in a place that is not abnormal, that just tells you that that normal part of the brain was capable of doing this all along. It has just been excited, right? It's been pushed into a certain region of behavior space where that sort of behavior is coming out. I think that's a very nice unifying sort of view to take. We don't take pathology as being something out here. It has nothing to do with the normal brain, which is over here. At least in the world of epilepsy, and Hughlings Jackson, who was the famous British neurologist who maybe almost started the whole field, said a seizure is an experiment, is nature's experiment, although a rough one, okay, that explores brain behavior. And those were the days where someone had a little shaking of the hand and they thought it was a seizure, and they thought, oh God, this must be happening in the motor cortex of that particular patient, right? So that is an experiment. That experiment tells you that there is a problem in one part of the brain that produces a symptom in some specific part of the brain, part of the body, right? So that is, I think, a useful thing. I mean, it's kind of unexpected, but it's worth holding in mind. And I will show you a little instance of that in another minute or two. If you want to be more poetic, maybe you can read Charles Sherrington, who wrote very evocatively about these things. And you might have heard about this enchanted loom metaphor, where you have patterns that are present, but they're always dissolving and they're always meaningful, says Charles Sherrington. Okay, so motifs that come and go. Okay, so how can we produce a motif? Can we artificially produce a motif? Well, I never want to, at least motifs that we consider not great for the patient, but sometimes we do without intending to. So this is a patient of mine from years ago. Sometimes what we do with patients when they're in a hospital bed with us and they're being investigated for their epilepsy is to electrically stimulate a little portion of their brain. That is done for medical reasons, which I won't go into, but they are FDA-approved devices that will transmit a small current between contacts within the head. And we do that essentially to excite that part of the brain, and we do that for more than one reason. This is part of the exploration, or the part of the overall, if you like, assessment of someone's epilepsy. Sometimes what happens when you pass an electric current, you might think that's a very dangerous thing to do. It can be if you choose the wrong parameters. We do it in controlled conditions. But sometimes what happens, despite your best efforts, is that current will produce a small seizure. That seizure lives really in the very place that you produced that electrical stimulus most of the time. Sometimes it'll spread. You get a small seizure right there. Okay, after you finish this stimulation, it ends in a few seconds. You don't really want them because they might spread and do the patient harm, and also they kind of confuse your own interpretation of the electrical stimulation. But anyway, sometimes they happen, and this is just an instance. And again, I just want to show you the very gross thing. You have a page of EEG here, 1, 2, and 3. All this business here is the stimulus artifact, as you're actually imposing this pulsating current in a very small portion of the brain. And then what you receive at the end of it in this particular situation is a motif that looks very much like a spontaneous seizure. It's called an after discharge, okay? And this is the way in which we can produce, not intentionally, but sometimes do, motifs that are spontaneously produced in the real brain, when a patient seizes. I thought this was a very nice instance of a motif that came out, okay? So a motif that was produced by a certain perturbation. And we've talked all week long about perturbations in various portions of the network at varying degrees of sophistication and various kinds of networks. And the fact that you perturb it in some way, and then you get a modular sort of organization that depends on where and how you perturbed it, right? Think of this as a sort of loose analogy to that. And you have here really a motif that appears because of some perturbation that you have executed. We've talked about the various types of oscillations that underlie the EEG. I wanted to understand this. I wanted to understand, in these very carefully controlled conditions, how is it you could produce a motif? What actually was different? What is the similarity or difference between the after and before? I have both the before and after conditions in front of me. I have a perturbing stimulus that I have applied, and now I have the emergence of a motif. And I'm just giving you one instance of how maybe temporal modules emerge, okay? I'm not saying, one of the open questions is how far one can take this kind of analogy or this kind of analysis. You might have heard, I'm sure many of you do cooperative phenomena, have heard of this very iconic model called the Kuramoto model, but it's really a phase oscillator. So you have an oscillator that's characterized by a unit amplitude and by a phase. So only the phase changes and the amplitude doesn't change. These are phase-coupled oscillators. And what am I trying to do here? I'm trying to say that the EEG is composed of these different rhythms. The intuition behind this whole thing is that the EEG is composed of different rhythms. After a perturbation, a few rhythms predominate. The others somehow just dissipate for a little while. And so some of them are selected. And you get the emergence of a motif that you wouldn't actually have otherwise, right? How might that occur? How do you suppress certain frequencies, and how do you enhance certain other frequencies? Well, this model tells you, and this was based on various other natural situations that had occurred, people have studied. Where you have an oscillator mix, this is typically quite large, 50, 100, 200, whatever, n. And each has its own intrinsic frequency. What are we modeling here in this very gross and rather simple but I hope illustrative way is to think of the entire EG somehow as being a summation, actually linear, just a simple addition, if you like, of oscillators that are intrinsic. We don't ask where their periodicities come from. We just take it as a given. And we say that these are all mixed and they have their intrinsic time scales. And over any piece of brain, they all coexist. And so if you take its spectrum of the entire composite signal, it's going to be broad. What happens after perturbation is that you get a monomorphic and you get a motif that has very specific oscillatory content to it or frequency content to it. And so what you might expect is for a spectrum to become compressed or condensed, where everything moves towards a common average, right? At least that will tell you. What will it tell you? It doesn't tell me about what's actually happening within the system. But phenomenologically, it tells me that what is happening is the selection of a particular frequency from what already existed. This goes back to what I was trying to say earlier, that a normal brain can seize too. And we often wonder, how come? How is it possible? Isn't seizure supposed to be bad brain? Well, it is. The bad brain happens to be in this part of the brain. And this guy is a bad influence on everyone else, right? And so he can make, or she can make, everything look bad, right? But if that influence goes away, then the rest of the brain begins to behave itself, right? So it's implicit. So see, here it is. The abnormal, the frequency that you finally get is implicit in the original. It is actually already there. And what you're doing is you're suppressing the others. And this can happen, for instance, through a model of phase coupling, where this coupling parameter increases sufficiently. As you can see, K is 0. These are just autonomous. They're all freely going around, doing their own thing. And here is a mean field sort of interaction term, whose strength is determined by the value of K. And when K goes past a critical value, you begin to get the entire thing behaving synchronously. The fact that this is just beyond a certain sort of superficial similarity, I think this is a little bit more than that for the case of after discharge, because this would predict the fact that the before and after spectrum were related like this, right? So that's one way in which we can actually just test this. You get something. This is real data, by the way. It's not a perfect Gaussian. This is the real world. But you see the before and the after. And I hope you're convinced that the after is a condensed, compressed version of the before. I'm going to move on, and we'll come back to the Kuramoto model in this last few minutes. We've talked about timescales of EEG. We've talked about how in the normal situation you have all this frequency mix. What we normally see is what are called the Berger bands, which is really a very small fraction of the entire variety of timescales that are present in human EEG. You get very high frequencies that we won't talk about when you sample finer. And you also get much slower oscillations. And these were a surprise to people who first observed them. And now the surprise has got even more, because you get oscillations at really, really, really slow frequencies. So if you have the EEG being collected over a space of a minute or two, you'll see all the Berger bands. If someone can keep the electrodes on their head for like a week, you might see a circadian fluctuation. That's natural. Okay, people go to sleep and they wake up. Now, if you do this for a month, you might get something that goes up and down maybe every 7 days. Now, that's a little more difficult to explain, right? Or you might get one that does every 30 days. Or if you have it for a whole, say, day, you might get oscillations that occur every few hours or every few minutes. So it seems as though if you extend the bandwidth, you get periodic time-dependent behavior that seems to actually have no limit almost, okay? At least limit in the sense, there's only so many months you can record this continuously. Some people have recorded this for years, by the way, but they don't see patterns that are any more than about a month or two, okay, in terms of their wavelength. So there's this question. So if there's a module, if if we say the brain is composed of spatial and temporal modules, or the spatial modules that produce their dynamics, and those are some characteristic frequencies that happen because of the way the ultrastructure is, then how do these slower oscillations actually arise? And why are they important, of course, before we even say that? Why do we bother? I'll go to this slide, okay? And forget, we'll come back to it also. I'll just tell you why. Because it seems when people do observational studies about these very slow oscillations, that seizures tend to occur at certain points in this cycle. So there's some sort of causative link between where you are, or correlation link, something that tells you that when you're on the upstroke of this, you might be in trouble. So isn't it useful to know when you're going to be on the upstroke? If this was roughly monthly, then you would say, hey, listen, it's the middle of March. Watch out, be careful in the middle of April, right? Don't speed on the freeway, okay? So that is the reason, why this might be useful. Might be useful to know, like where, by what mechanism is this occurring? Is it possible to perturb the brain so that this, say, becomes less in magnitude, or it goes away altogether? Where do these rhythms arise from? So where does this other extra added temporal modularity, something that seems to be superimposed on the original temporal modularity of the EEG, arise from? We observed this, by the way, and I'll just fly through the next 3 or 4 slides and get to the end. This is work with Sai Bhavisetti, who's been, by the way, an excellent collaborator. I have to thank Reinhard for introducing to this very fine person who has taught me so much, and we have worked very effectively together. And what we did was construct network matrices of patients, and the details aren't important. It's just the fact that if you compute, for instance, and we took several sort of metrics, but we were interested in whether this network was disjoint or connected. And you take, say, a measure of this, which is the Fiedler eigenvalue, I believe, lambda 2, or the spectral gap. It tells you how far away you are, or how connected the system is. And you see that that entire lambda 2 varies as a function of time, and varies over time scales of minutes. So it's as though the whole thing is there is some sort of dynamic synchrony to this, and that dynamic synchrony is oscillating at time scales that are very much slower than the actual EEG. Now, isn't that important if we think of epilepsy generally being something, or engendered by something that's all together? So maybe we should understand that dynamic. We should understand where that comes from. These authors took it upon themselves, and they actually went back to Kuromoto. I was pleasantly surprised. So this is really a big favorite, and virtually every, or many attempts at modeling cooperative behavior in multi-oscillator systems will hark back to this particular formulation, where they took this very thing that which I showed you earlier, with a slightly different sort of formulation, where they took where they tried to model the emergence of these slower oscillations from, in fact, arising from an interaction between the circadian oscillator and some imposed external forcing term, and that forcing term was of the period of a day. Okay, so it's a circadian Zeitgeber, right, which is a roughly 24-hour external forcing term. And essentially you get, they're able to say for very specific parameter values. We wanted to explore this in our work group, but as like an exercise. For certain specific parameter values, I think of what alpha and epsilon maybe, you get these very slow fluctuations, and intuitively they emerge because of some kind of interference effect between your natural circadian rhythm and the Zeitgeber that is sort of pushing and pulling at it. I call this infrared EEG, by the way, to distinguish it from ultraviolet EEG at the other end. Okay, it's not a widely used clinical term. Okay, so I will just go right to the very end. I just want to say there's a vast number of phenomena, and I've not really described several of these. It was my plan to do this, but I just restricted myself exactly to what I thought I knew best, which was epilepsy. We have talked a little bit about dynamics, the phenomenology. We've talked about a little bit about the changes in morphology that are given to us. That's just the way nature made it. There is a different question about how they arose. We're not going to question that. We just take this as a given. we recognize the presence of timescales, and we ask the question, how many of these are truly fundamental, and how many of these can be derived, right, from self-interaction? We've seen at least 2 or 3 phenomena. There are dozens more, scores more. Okay, flip-flop switches that occur in the whole brain, flip-flop switches apparently in the dolphin, okay, that go from state to state, hemisphere to hemisphere. There are other kinds of microstate trajectories. I think maybe Danny Bassett, mentioned them as you go through. People have described the fact that, the distribution of power in the EEG tends to go through some sort of 4-cycle. They call them microstates. Not clear what produces them. There's a huge literature, of course, on the formation of dynamic patterns. You would have heard of grid cells, which are related to navigation, first found in the rodent, but now have been discovered in the human too, or at least have been described. And of course, we have seen seizure in a little bit of detail, and maybe 1 or 2 examples of the whole zoo of pathological patterns of spatiotemporal motifs that we see in our own world. There are a whole lot of other models. There are a whole lot of models that have been brought to bear in some of these problems. We've looked at the Kuromoto in a little bit of detail, but not very much else. All of these models can be put on networks, of course. And those networks might themselves, of course, have modularity. So there's, a lot of formalisms available to us. But again, I guess I have to now quote Laubenbacher here. This is not published, but this is personal communication. So he said in an email to me, please talk about what modularity is good for. Why should we bother? Okay, I guess that's what you meant, Reinhard. Okay, so this is my explicit instruction, okay? So, well, I think, you understand data. We fortunately have problems that, appear interesting. Those problems tend to arise in patients who we want to make better. It is possible that understanding these phenomena will eventually improve patient care. So there's a certain motivation there that is very practical. So, so these are all the ways we can understand the data by themselves. Of course, we can relate that to functional anatomy. We can understand even things that really right now are still open questions. Why do seizures occur when they do? What is the role of these ultradian or multidian rhythms, etc. Perhaps, that will tell us a little bit about how to treat things or what targets to go after. And then finally, and much more speculatively, of course, modularity may be good as a brain theory, or might inform, or it might itself be a brain theory. Okay, I'll just end there with, I thought Warren Weaver's statement was very good, and I think probably describes the flavor of this workshop of bringing people from diverse groups. But this is our group, and all we do is look after patients with seizures. But you can see just in the variety of people what it takes really to create something that's an effective, we hope, clinical unit. So with that, I'll thank you.
Son. 1, 2. Yeah, now it is. Okay. Great talk. Thanks a lot. I don't know much about the field, but one question that was bugging me from the beginning is, are there seizures detected in simpler animals? It seems to me like what is the simplest system where one has detected seizures? That's one part. And the other part is unrelated. It's a very general question. I've heard some groups have looked into criticality and epilepsy seizures, right? And I just was wondering what you had to comment about the relation between that and what you said.
I'll answer the second question first. I don't know. I've heard this is Beggs and Plens from the NIH, right? Mm-hmm. So they do, in vitro work, and they look and they claim that their work goes back 10 years more, etc. And it is thought that maybe it's not seizures as much as the mode of how the cortex behaves. Apparently, it drives itself to the so-called edge of chaos, where it has a lot of diverse behavior available to it. Mm-hmm. And that is a natural place for it to gravitate to, given all the demands that there are computationally. There was interest in trying to map some of these or try to find evidence. There's a whole literature on criticality and EEG. I don't believe that hypothesis is and actually we've published on this too. The models of EEG, often they'll say it's a 1/f. There's no reason to assume that. Yeah, and a 1/f will fit it, but so does a whole lot of other functions. And 1/f, of course, is monotonic. It doesn't have the actual peaks in it that we observe. The alpha rhythm is an actual peak. right? So it doesn't explain the continuum that the EEG actually is. So I have difficulties with some of that. But again, on the in vitro, down at some other level of observation, that might be a very useful thing. So I don't really have an opinion, I should say. But of course, your other question was, what is the critical size? Yeah, people do. And again, there are whole groups and people who spend their careers looking at seizures in simpler systems. Zebrafish, I believe, is the simplest system I know of that has been thoroughly studied or is under extensive investigation for seizure. But they may be simpler animals still. And of course, there's a great advantage to understanding these simple systems. Right?
Thank you.
Next question. Will be maybe pass it along. I think he had a question too.
Hi, thanks for the great talk. I have one.
Is the microphone on?
Hello.
Yeah.
Yeah. Hi, thanks for the great talk. I have one question. So you define this module as a spatio-temporal motif, which is like a kind of predominant pattern in the signal.
Yeah.
So I'm assuming that there will be some compositionality, like all the bands are formed of this kind of patterns, right? And it's also a kind of like every seizure is producing different, like every time it's producing different kinds of patterns. Is it
Not really, no. It usually tends to be the same pattern, the same type. I mean, so it's the same phenomenon that occurs repeatedly. Okay. It's not just a subjective symptom. The patient says the same thing is happening to me that happened yesterday or last month or 2 years ago. But actually, when you look at the brain waves, they look the same each time. The disturbance that happens is very stereotypic. So it's accessing the same state each time.
Okay. So here the compositionality means that pattern, its whole signal is composed of that patterns. That is what you mean by
That's right. Yeah.
And that has a meaning.
That has a significance.
Explain
that has a certain significance, right? And that is abnormal or extensive enough for it to actually perturb the patient, clinically, and that causes symptoms and so on. Yes.
Thank you very much. Okay.
Yeah. So I was wondering, in patients with epilepsy, of course, you notice very significant differences in the EEG when they're having a seizure. Are there characteristic differences?
There are.
When they're not having
there are. Yeah. So those differences, it's a very good question, and that highly practically significant, I should say, because let's say someone has a big seizure, and then one of the problems we always have is this something that has actually basically started from one place and gone to another? And how do you even know that when you're observing the state where it's fully developed, right? There are many ways of trying to get around that issue, but one of the things that is directly relevant to what you said is that even when they are not having a seizure, that particular part of the brain looks a little bit different. Now, the spatiotemporal motifs, if you like, it exhibits are different and correspond to whatever ultrastructural or whatever local connectomics that is disordered, constitutes the particular pathology. And so from that spatial module, you get a different kind of baseline temporal modularity, right? You get a different kind of time-dependent behavior. I mean, you get a different appearance. The EEG itself looks different when you're not in the seizure. But then from time to time, this thing will enter a state where then this particular state then goes and gets transmitted to other portions of the brain.
I enjoyed the talk, Giri. The question here is, I have two questions actually. The first one is looking at the Kuramoto the equation. Can you add a perturbation term from something like a vagus nerve stimulation therapy, and say, okay, this is going to predict this specific changes in the time?
Okay. Do you have another question, or can I just answer that?
Second question is, I'm interested in ion channels and like ion related mishaps, like for example, like potassium currents and calcium currents being different and hyperactivating neurons and stuff like that. How can we, so in epilepsy, for example, how can we sort of get to a stage where we can sort of decouple the individual currents, and so maybe make it modular in ions and ion channel variability and stuff, and maybe somehow tackle the problem with specific ion in mind?
I thought that's what you did with voltage clamp. Is that not correct? Yeah. So yeah, you do decouple the ions, experimentally with techniques that, as you know, are very well established. Generally, when you have channelopathies that cause epilepsies, you will have abnormal conductivity, sometimes channelopathy, potassium, sodium, etc. And as you well know, many anticonvulsants actually act exactly at that level, act at sodium channels or potassium channels, etc. This, by the way, is a very macroscopic model. Remember, the ion channel fluxes are a very different phenomenon, and they have no sort of ensemble type description to them, right? So this is what is happening in a localized piece of cell membrane. Ultimately, of course, that's relevant, but that's at a different level of organization. In terms of Kuromoto being actually applicable, I think it's an allegory. I think it's a useful one. I think it gives you a certain prediction, which in a loose way, some sort of overall gross way, we can prove, okay, that something of this type is happening. But the question is, what is it that's causing the increase in K in that particular parameter? So what is the neurophysiological instantiation of that? I don't really know. I mean, you can speculate, but then again, that will probably be mediated by some neurotransmitter, something, the GABA is depleted, a glutamate is increased, or something like that. But, I don't have that data. I'm not an experimentalist. It would be very nice to know.
Last question. Last question. There's a little delay. There you go.
Giri, thank you for this perspective from a potential end user of an operational modularity theory of the brain. The question I have is, you showed us how you collect these data that you showed us, which is by placing probes in one area of the brain. So that will give you a very partial view. Are there methods to collect data from the entire brain?
Yeah, we do that too. Clearly, you can't put probes, these kinds of probes everywhere. So the fact that you have something that can be collected from the outside is necessarily non-invasive. It's it's done from the surface. So you do have other surrogates of that, okay, which have their own advantages and disadvantages. But yes, we do have whole brain recordings, and we often have them in the same patient. And in fact, we're obliged to have them in the same patient. Otherwise, you wouldn't know where to put the electrodes that go in. So yeah, we have, finally you have like a whole lot of data points and a whole lot of situations where data has been collected. I'm just highlighting one stage where we are in the process of data collecting to understand the patient, what kind of things that we see. But our, of course, ability to collect data in the ideal situation is limited. These are real patients. But I think we see enough for us to be able to, I don't think we are actually hampered by a lack of sort of, if you like, comprehensiveness. I don't think so. I think there's so much in here that, can be answered with the data we have already. So it depends on the modality is the answer. I mean, so we do have whole brain. Of course, we have the scalp EEG I showed you earlier, it's the whole brain. But the invasive electrodes, which will give you a more fine sampling, necessarily in a small area.
And I have another really naive question. You showed us this picture where you see a pattern of very little activity and then a lot of activity. Not knowing anything about anything, I wouldn't know which one of these two is good and which one is bad.
They're both bad.
They're both bad.
Yeah, because you're not supposed to do that, right? So neither of those patterns are actually seen in healthy people. So you see, so yeah, that's right. And then eventually when somebody recovers, that alternation becomes much less obvious. And then the quality of the rhythms itself begins to look like what the very first EEG I showed you, which is, the normal thing. But as in sickness, you see this. No, everything is bad.